His laboratory focuses on the study of beneficial bacterial molecules from the human gut microbiome as novel therapies for immunologic and neurologic disorders, with a specific focus on developing probiotic treatments for autism. He's a founder of 2 biotech companies and has or currently serves on the scientific advisory board of over a dozen companies, academic centers, and not-for-profit foundation. I would like to welcome Dr. Mazmanian. He's also the scientific advisory board on the scientific advisory board of the Brain Foundation and a great help to us. Thank you so much. Take it away.
Thank you, Pramila. While I still have you, I assume you can see my screen and hear me.
Yes.
And thank you for that wonderful introduction and also for inviting me again this year to speak and participate in chairing this session. I pull up my slides. I really enjoyed the support from the Brain Foundation for our research, as well as the network that you're building of scientists for us to collaborate and really think about pushing the scientific boundaries of this really obviously interesting scientific area. So I'll chair this morning's session, which is loosely themed basic and translational research in ASD. And I'll start by discussing some of our work, give an introduction to a project, and then the second half, talk about some newer work that is being funded by the Brain Foundation. And almost everything I'll present today is unpublished. And so obviously the microbiome is familiar to the audience and was discussed yesterday, but just roughly speaking, we have 100 trillion bacteria living in and on our tissues throughout our entire lives. They contribute to the genetic material in our body. In fact, many people have termed the microbiome the second genome because there are actually 150 times as many gene families in our microbiome, which is the collective genomes of our gut bacteria, than it is in our own genome. And so the 2 genomes essentially are working together to mediate the balance between health and disease. And the bacteria themselves have the metabolic capacity in aggregate of the liver and weigh approximately the same as a brain. So it's this foreign organ that we all harbor in our bodies that we believe critically contributes to our biology and to our health. And it's been shown, and I'll just go quickly over the disease areas. It's been shown that the microbiome is related to a number of different disorders and conditions, many of which have been done in animal models, and I think that that's still where the field is. That though a lot of work is being translated, and again we heard some of this yesterday, the vast majority of the discoveries and the innovation that is coming in the field still happens in animal models. I think that's reflective of, again, the fact that this is a new research area, but also, there are still tools that are being developed and other technologies and even regulatory aspects that are being put in place for an increase in human research in the microbiome. I think we need that, and there's more coming. Just loosely in terms of how the microbiome can interact with the brain, there are a number of different conduits that include the production of neurotransmitters in the gut under the regulation of microbes, use of the immune system, where most of the immune cells in our bodies are in our gastrointestinal tract at any one given point in time, and they're educated or primed by gut bacteria, and then these immune cells or their products such as cytokines can circulate throughout the body and even have been shown in animal models to mediate behavior. Neurons can have receptors for these immune cytokines. What I'll talk about today is this class of communication where molecules directly produced by bacteria can enter the circulation, in some cases get into the brains of animals and modulate physiology, brain activity, and behavior. And there are obviously neuronal connections as well that link the gut and the brain. And it's really important to remember that there's a lot of evidence that the communication is bidirectional. There's signals going from the gut to the brain, and then from the brain to the gut. We can all think about this in our own lives and day-to-day occurrences of how maybe when you're stressed in terms of your gastrointestinal tract, then it may, you may have difficulty in thinking or sleeping, or if you have anxiety or depression, something's on your mind, it may affect your GI tract. Again, those are just broad examples of the connection between these two organs. But experimentally, there's still a lot more work that's been done in looking at gut to brain connections, just because it's technically easier. And I would argue we still need many more studies where the trigger or the inciting signal is in the brain, and we need to understand what's happening in the gut. But again, it's bidirectional in communication. But today we'll focus on molecular signals that emanate from the intestines and affect brain function. And so a few years ago, I won't give you the background for this paper. Obviously, it's been published quite some time ago, but we showed in this manuscript that the microbiome is altered in a mouse model of neurodevelopment. This is the maternal immune activation, or the MIA model of neurodevelopment, which has many features of autism and schizophrenia. And in addition to an alteration in the microbiome that was coupled with behavioral changes, we found changes in the gastrointestinal tract, both in terms of epithelial barrier dysfunction as well as inflammation. And then wanted to understand if there's a link between the gut and the brain, just as I described in this previous slide, and then performed an untargeted metabolomics analysis. And what that essentially means is we looked for small molecule differences between mice that had behavioral symptoms consistent with autism and mice that did not. And so again, I know I'm not providing all the background for this, but because I want to get into the unpublished work. But very briefly, these are animals that do not show features of autism. These are animals that do show features of autism, and I'm talking specifically about behavioral features. And you can see that in the animals that have behaviors that are autism-like, that there's an elevation of this metabolite called 4-ethylphenyl sulfate, or 4-EPS. There are other molecules that were dysregulated, but we focused on 4-EPS, almost a 40-fold increase in this one metabolite, which was the highest change in the entire data set. And so over the past few years, we've been trying to understand what the function of 4-ethylphenyl sulfate is. And what we suspected, and we now know, and I'll show you data for this in a minute, is that bacteria can convert dietary tyrosine, excuse me, to a number of different molecules, one of which is 4-ethylphenyl. This happens in the microbiome, this happens in the intestines. And then when 4-ethylphenyl enters into the circulation, we believe it's very rapidly sulfated to 4-ethylphenyl sulfate, presumably in the liver, though this reaction can happen elsewhere. And then ultimately, the molecule that is in circulation and even in the brains is a metabolite called 4-EPS, the sulfated version of 4-EP. And I also want to emphasize that though I'll talk exclusively about the biology of 4-EPS today, we really think that this is a harbinger of information for additional metabolites. There are many dozens of metabolites that fall into the same category. They're either tyrosine or tryptophan metabolites, but they're these phenolic molecules produced by the microbiome, which in many cases seem to co-vary with each other, meaning that they go up and down in the same individual, and presumably have biological functions that are either similar, overlapping, synergistic, and need to be studied. So again, I'll talk about one metabolite, but there's many, many more metabolites and combinations and reactions that we believe are happening in vivo. And so, as I just presented, we discovered this elevation of 4-ethylphenyl sulfate in this animal model, in a mouse model of neurodevelopment. And the reason why we discovered it was because it was in a particular panel of chemical standards for an untargeted metabolomics analysis done by mass spec. And what had happened is that there had been, previous to this, a number of different metabolomics studies in humans, but they just weren't picking up this molecule. It may have been in the biological tissue, whether it's urine, plasma, or the tissue that we looked at, but it had not been reported. And so we went back and addressed this issue. So working with Paul Ashwood at UC Davis, we were able to obtain samples from a charge study that had run there many years ago, where they had collected plasma and feces from a large number of individuals with autism and typically developing individuals. And so this was obviously interesting to us because very, very few studies, even to date, have collected feces from individuals with ASD. And so we did a metabolomics analysis. There are many, many metabolites that were changed. This is a paper we published last year, but I'll just draw your attention to 4-EPS. There's a subset of individuals between 30 and 35% of the ASD population, at least in this cohort, which had these very, very high levels of 4-EPS compared to individuals that were in the typically developing group, or samples from individuals in the typically developing group. So it's not all individuals with ASD. I mean, the metabolite is detectable in all individuals in the study, but then there's this subset where the elevation of this metabolite is quite high. And then if we think about what smaller subset of individuals show the greatest increase or fold increase in 4-EPS, it's in those individuals that have an ASD diagnosis with GI issues. And so individuals with ASD without GI issues have about 5.5 to 6-fold increase. Those individuals with GI issues have over 9-fold increase. So again, more suggestive that there may be issues related to the gastrointestinal tract in this metabolite. And so we wanted to, again, understand the function of this metabolite and go beyond these correlations. Of course, this metabolite and or other metabolites can serve as potential biomarkers for disease progression or for potentially even interventions. But we want to understand, is there a physiological or biological function to these metabolites? And so obviously we start as typical in biomedicine, we start with animal studies, because these are proof of concept studies to just convince us or to demonstrate that there are indeed physiological changes relative to this metabolite. And so we've done a number of experiments where we've fed the metabolite, we've injected the metabolite. I won't talk about that data, but it's all consistent with what I'll show you. But working with Michael Fischbach at Stanford and his very talented postdoc Masa, who essentially deconstructed the biosynthetic pathway for conversion of tyrosine to 4-ethylphenol, which I just alluded to. And so what Michael and his team discovered was that there are 3 enzymes, all bacterial enzymes, that can convert tyrosine to para-coumaric acid, which is then converted to 4-vinylphenol and then ultimately to 4-ethylphenol. Again, this all happens in the gastrointestinal tract. The system can use para-coumaric acid or p-coumaric acid as a substrate as well. That's found in certain dietary compounds in certain plant-based foods. But the system appears to prefer to use tyrosine, which, as many of you know, is an amino acid that's found in diet, and also we get from our bacteria as well. And then what Michael did was that he engineered bacteria to either produce or not produce 4-ethylphenol. So the exact same 2 strains of bacteria, Bacteria ovatus and Lactobacillus plantarum, that either do not have the genes for 4-ethylphenol production or do have those genes. And so if they do have the genes, these are 4-EP plus bacteria, and then we call the same exact organisms, just missing 3 genes, the 4-EP negative bacteria. And so again, 2 bacterial strains for each pair that are different, to the best of our knowledge, in the production of this final metabolite 4-EP. And so we colonized notobiotic animals with the 2 strain pairs or 2 sets of bacteria to produce mice that either do not or do produce 4-ethylphenyl sulfate. And again, this is a reductionist system, but it's a very clean system in the sense that we know that all the biological activity that we observe in this animal is dependent on the production of 4-ethylphenol in the gut. And so we show that to indeed to be the case, is that these bacteria do produce 4-EP in the feces if they contain the genes for conversion of tyrosine to 4-ethylphenol. When we look in the urine, and we see the same data in the blood, that there's a high level of 4-EPS in the circulation. And very interestingly, we can detect almost no 4-EP. So it's a very quantitative conversion of 4-ethylphenol to 4-ethylphenol sulfate. But again, we can see levels of this metabolite in the circulation. And we can also detect 4-ethylphenol sulfate in the brains of these mice if the bacteria can produce 4-EP in the gut. And so the metabolite reaches the brain. We don't know if the final function is in the brain, but it's interesting that the metabolite is indeed there. And so we did a number of studies I won't talk about for the sake of time, where we looked at brain activity and brain connectivity, and we see some very interesting changes in particular regions of the brain that are more or less activated in the animals that were exposed to 4-EPS. And then we wanted to understand if we can explain those brain-wide changes that I just described at the molecular cellular level. And so working with Dan Geschwind at UCLA, we performed an RNA-seq experiment for several different brain regions in each of the 2 cohorts of mice. And almost all of the genes, or the vast majority of the genes, over 75% of them, that were dysregulated either up or down at the transcriptional level on the paraventricular nucleus of the thalamus, or the PVT. And you can see that that's here in green. Many of the genes are found in neurons. So when we do cell-specific enrichment for these transcriptional pathways, we can see that there's certainly changes in the neurons, but also it looks like there are changes in these cells that are various different maturation states of oligodendrocytes. So OPCs are oligodendrocyte precursor cells, NFOs are newly formed oligodendrocytes, and then MO is mature oligodendrocytes. I'll talk about oligodendrocyte biology in just one second, but we wanted to validate to see if indeed the genetic signature that was being picked up by the RNA-seq was giving us the insight into oligodendrocyte biology. And so there are a number of genes associated with oligodendrocyte precursor cells, and these genes in green are all elevated in the 4-EP positive mice. There are a number of genes associated with mature oligodendrocytes. All of these genes are reduced in their expression level in the 4-EP positive mice. And so this really gives us this picture that there is, it appears at the genetic level that, or the transcriptional level, that there's this arrest, if you will, in maturation of oligodendrocytes. They seem to be stuck in this OPC state, generally speaking, and reduced in their efficiency to convert to mature oligodendrocytes. At least that's what the RNA data told us. And so we wanted to now validate that to be the case. So using immunohistochemistry for particular markers that can differentiate mature and immature oligodendrocytes, we showed that indeed the mice exposed to 4-EP, so the animals that had 4-EPS in their brains, have less mature oligodendrocytes when we use, again, this marker of mature oligodendrocytes CC1 over NG2, which is a marker of oligodendrocyte precursor cells. We've used a number of other markers as well, again, not shown here for the interest of time. But importantly, the number of oligodendrocytes in total is not different between the two. So we don't think there's an issue in terms of development of oligodendrocytes, but some likely dynamic delay or arrest in their maturation. And this is again by immunohistochemistry. We can validate this by flow cytometry as well. So these are the mature oligodendrocytes that are found in abundance in the 4-EP negative mice, and they seem to be missing in the 4-EP positive animals. And so again, this is quantitated here. So the function of, so again, all the data suggests that there is either an arrest or some sort of halt in the maturation of oligodendrocytes in the brains of these animals. It is region specific. It is certainly found in the PVT and other regions as well. And looking at the subsequent effects of this delay in maturation, again, as many of you may know, oligodendrocytes have a number of functions, primary to which are the laying down of myelin around neuronal axons. And so myelin helps conduct signals across axons over long distances. So think of this as like the coding on a copper wire in your home. And there's increasing evidence that certain behaviors and different neurodevelopmental conditions are associated with changes in myelination patterns in the brain. And so having seen this effect at the cellular and transcriptional level in oligodendrocyte maturation, we wanted to see, is there a change in myelination in these animals? And so initially looking at the corpus callosum, where the neuronal fibers are aligned, so it's very easy by electron tomography, which is what we're looking at here, to physically quantitate the degree of myelination as well as the thickness of myelin. We can see that there are many more unmyelinated axons in the 4-EP positive mice compared to the 4-EP negative mice. And when we do analysis called the G ratio, which takes into account the variable shapes of these cells, we can see that the higher G ratio, which correlates to thinner myelin, is found in the 4-EP positive mice. And then when we actually just look at the thickness of myelin in these tomograms, we can see that the myelin is thinner in the mice exposed to 4-EPS. So again, consistent with the arrest in oligodendrocyte maturation. And then more specifically in the PBT, here we used functional ultrasound to look at the organization of myelin in the structure. We looked brain-wide as well, and we see very similar effects, that there's just more disorganized myelin in the 4-EP positive mice compared to the 4-EP negative mice in this particular region, again, where we saw that strong genetic signal. And then ultimately, we wanted to now correlate this back to behavior and ask, is 4-EPS altering behavior in these animals? So we performed a wide variety of behavioral tests. Here I'll show you the open field test, where animals are put into an arena, and the more time they spend against the walls, the more anxious those animals are, the more they venture into the center, the more bold those animals are. And the animals exposed to 4-EP in the gut, which again have 4-EPS in their circulation in the brain, spend less time in the center, more time at the wall, suggesting that they have an anxiety-like phenotype. There's no change in locomotion, which is an important control, because if the animals were sick for any reason, it would affect this outcome. In fact, we've done an extensive amount of analysis on the health of these animals. We see no health differences in any parameter. We've looked at GI health, brain health, weight, immunologic inflammation, and a number of other parameters, and really can't find any signs of overt disease or illness in these animals. They seem to be perfectly fine, but they seem to choose not to enter the center. And this is supported by data from the elevated plus maze, another test for anxiety, where animals that spend time in this open arm are just less anxious and more bold, and the more timid animal will spend time in the closed arms. And you can see that the mice exposed to 4-EPS should spend less time at the terminus of the open arms compared to the 4-EP negative group. We've actually shown in 2 additional tests that these mice have anxiety. So that's a very, very strong phenotype. Excuse me. But we also wanted to look at ASD-like behaviors. So we looked at social interaction in the direct social interaction test. There is a non-significant decrease in social interaction, which is consistent with social deficits, in this peculiar change in anogenital sniffing, which I'm not sure we can fully understand, but is a known behavior of animals. When we looked at ultrasonic vocalization, essentially how these animals communicate with each other, there's a decrease in the duration of each call. So every time they emit a call, their sentences are shorter, if you will, as an analogy. So these animals appear to have changes in emotional behaviors that are consistent with anxiety and with autism. And we did motor test these animals. We found no motor deficits. We tested the animals for cognitive learning and memory tests. We found no changes between the 2 groups. So it doesn't look like there's a gross change in behavior in these animals. It seems to be more confined to emotional behaviors. And so just to summarize this project and this preclinical work, either changes in the gut or the products of the gut known as the metabolome, small molecule products known as the metabolome can be different, either in animals and potentially in humans. And then this leads to a change in the flux of microbial metabolites across the gut barrier. And in the case of 4-EP, 4-EP is elevated and can then be sulfated in the liver, and essentially its concentrations increase in the circulation. And the metabolite does enter the brain, and it leads to an arrest in oligodendrocyte maturation, which then subsequently leads to a reduction in myelination, both in terms of the number of axons myelinated and the thickness of the myelin itself. And then that alters behavior. We have some functional data that does correlate the myelination and the oligodendrocyte phenotypes to the behavior. So we really think it's working through oligodendrocytes. And so, so this work, obviously, we're interested in identifying what are the receptors for 4-EPS, what are the specific cell types that are being directly impacted by this metabolite, and very importantly, to learn the function of the many other metabolites, as I mentioned, that we believe are in this same chemical class and may have biological functions as well. And so Stuart Campbell later this afternoon will give a talk on how this work has been translated into the clinic. So again, we run a basic science laboratory, but Axial Therapeutics where Stu is the CEO, has taken this technology into the clinic. And again, I urge you to listen to this talk. And so we, all the work that I just showed you has been done in wild type animals, but of course, autism is a genetic disorder, and we want to then understand what are the effects of either the gut or the microbiome in the context of ASD-related genetics. And so we started working with the SHANK3 model. And so what SHANK3 is it's a gene that's believed to be the cause of Phelan-McDermid syndrome, whether it's through deletions in 22q13 or mutations that lead to haploinsufficiency. Insufficiency leads to Phelan-McDermid. And most individuals with PMS have ASD. I think over 70% of Phelan-McDermid individuals have autism spectrum disorder. And then they have a range of different behavioral and physiological features, which do include gastrointestinal and elevation of gastrointestinal issues. What SHANK3 is, it's a scaffolding protein in the postsynaptic density of excitatory neurons, and it has an effect in glutamate signaling. It's been widely studied. Again, I'm sure many of you know the literature on SHANK3, and of course, Joseph Buxbaum is an expert in this area, and he'll be speaking next. And so again, we wanted to see if there were any microbiome or gastrointestinal effects in the SHANK3 model. And this is there is a mouse model that had been developed several years ago, where it's a deletion of SHANK3, which results in several behavioral changes in the animals. This decrease in rearing in the open field. These animals groom themselves excessively. So here you can see the knockout animals, SHANK3 knockout animals, excessively groomed to the point where they have these lesions, and then they have an anxiety-like phenotype as well. And so there's a well-established mouse model. There are even non-human primate models for SHANK3. And it had already been shown that the microbiome is related to the SHANK3 animal, in the sense that when people had compared wild type to SHANK3 mice, that there's a decrease in Prevotella and Lactobacillus species, and then supplementing SHANK3 animals with a particular organism, Lactobacillus reuteri, changes their behavior in the social paradigm in the 3-chamber social test, as well as changes in the receptor for GABA, in particular brain regions. And so again, there's emerging evidence the microbiome plays some role in the physiology and the behaviors of this metabolite. There's an unpublished, I believe still yet unpublished bioarchive paper from Drew Carrilli at Mount Sinai, who Joseph works with, that has shown that antibiotic treatment appears to exacerbate symptoms in this mouse model. So just very quickly, these are the knockout animals with antibiotics, and they spend less time with another animal compared to an inanimate object. So they show the social defect, which is exacerbated over the animals that weren't treated with antibiotics, and that's quantitated here. And then same in the open field, there's an anxiety phenotype that is increased upon antibiotic treatment of the knockout animals. And there are also changes in gene expression in this paper as well. And again, here's the reference for that work. So there's preceding evidence that the microbiome is both altered and appears to impact behaviors and other outcomes in this animal model. And so Jessica Griffiths in our lab is interested in what's going on in the gastrointestinal tract of these mice, and has performed a whole gut transit assay, where she essentially feeds a dye to the animals and looks at how long the animals take for them to pass this tracer, if you will. And the gut transit time, in terms of the whole gut, is much faster in the SHANK3 animals compared to the wild type animals. So essentially, there's a faster movement of contents through the entire gastrointestinal tract in the SHANK3 knockout mice. And when she did an experiment where she isolated the effects of just the small intestine, she wasn't able to see a difference. So there's a different diet and a different protocol to ask, is there an effect on gastric emptying or in small intestinal transit? And she does not see a phenotype there that would explain that. So what we think is happening is that there may be a change in the colon that results in this effect. And so what Jess did was she set up an ex vivo system where we can take colons of animals and put them in this organ bath. and then using a video tracking software, look at the contraction of the muscles in the colon and ask, are there differences between mice, between the genotypes, is what you've done here. But the system also allows us to flow neurotransmitters, other metabolites and molecules through the system to see if gut motility has changed. And so in this CMMC assay, and this is live tissue, which you can see here, the data show when there are contractions based on the width of the tissue. When there's contractions there, the gut tissue is narrower, and you can see that in the red. And indeed, in the colons of the SHANK3 knockout animal, there are more migrating complexes, so more muscle contractions, if you will, in the knockouts compared to the wild type, which likely can explain the whole gut transit data that I showed you previously. So we're obviously very interested in why knockout or deletion of SHANK3 in the colon appears to result in this change in contractility. We're following that up. I should mention we have animals where we can selectively knock out or express SHANK3 in the gut and the brain. This is also funded by the Brain Foundation grant. And we wanted to understand, are there physiological changes if we just have SHANK3 in the brain or we just have SHANK3 in the gut, and how that affects either GI or behavioral functions. And those are ongoing studies. What just wanted to then understand is there now a susceptibility in the colons of these animals to insult? And so we used essentially a colitis model, the dextrans sodium sulfate model, the DSS model, to ask, are there changes in essentially inflammation or colitis. And initially you can see that the SHANK3 knockout animals are more susceptible to DSS colitis. They lose weight compared to the wild type animals. And then when we look at the colon, they have this hallmark shortening of the colon, which is consistent with not just mouse models of colitis, but of human ulcerative colitis or Crohn's disease as well. And so essentially the intestines of these mice are just more vulnerable than the wild type mice. And we've also become quite interested in the microbiomes of these animals. So I showed you a few slides ago, there were 2 studies that have shown the microbiome is different, but we know the microbiome is different for animal models in general, because commercial mice just have very depleted or reduced microbiomes. And so this is actually reflected here from this recent paper. These are the microbiome profiles of one of the 3 large commercial vendors. And as you can see, these microbiomes really are diminished or reduced in the diversity of the microbes that they harbor. And then these are microbiomes from animals, wild mice caught in 3 different locations. This happens to be in Maryland. And you can see how feral or wild animals just have much more diverse microbiomes. And what's really interesting is if you take the same laboratory strain of mice and then colonize them with either wild microbiome and compare them to the commercial microbiomes, you see a very dramatic difference in the immune response. I mean, it's pretty remarkable to what degree the immune system of these animals are shaped by the diversity of the microbiomes in them. And this has been done for a number of other papers, but clearly you can see here that the wild mouse microbiome just has a higher pro-inflammatory response than that of the laboratory mice. So I want to see if this principle applied to neuroscience. So we took both the wild type and the SHANK3 knockouts, and then we essentially diversified their microbiomes by adding these wild mouse microbiomes. And what you can see, some really interesting features, is that just in wild type animals, there's this increase in I should explain the behavior test. This is the O maze, zero maze, where it's very similar to the elevated plus maze, but instead of being a plus, it's a 0. And so here are the animals where the animals feel safer because it's in this closed arena. Here's the open arena where the animals are less anxious if they spend time there. And clearly the animals that were colonized with the wild microbiomes are less anxious. And we see this really interesting, reveals this really interesting genotype effect in the wild R microbiomes if the SHANK3 protein is knocked out. And so this is in males, and we can see less of an effect in both sexes, but clearly the wild microbiome is changing the behaviors of both the wild type mice as well as the knockout animals. And that's true in the open field as well, but here the effect is more pronounced in males and females, where you can see that the animals that have the wild microbiome that that were knocked down in SHANK3 spend more time at the walls, which is where they feel safer, meaning they're more anxious compared to the wild type mice. So obviously more studies coming, and we wanted to understand how this affects other behaviors, how this affects GI function. But I think that there is a lot of biology that we're missing in the way that we house and rear laboratory mice, and some of that is revealed by just diversifying their microbiomes to be more representative of what an animal would look like in nature. And so I'll just leave you with this concept, just at 30,000 feet of the balance between genetics and environment in studying autism. And I think oftentimes the role of environment and the role of genetics are studied separately. In other words, people look for genetic etiologies, other groups, other laboratories, other teams look at environmental etiologies. I think there's just a lot more work that needs to be done at the interface to look at gene-environment interactions and to see to what degree those may explain outcomes in animal models, and certainly, most importantly, in people. And I'll just finish by thanking Brittany Needham, who did all the work I showed you on 4EPS, and Jessica Griffiths, who's a graduate student in our laboratory who led the SHANK3 project. These are all of our collaborators on the work that we do in our laboratory. Here are our funding agencies, and huge thanks to the Brain Foundation. So I'll stop there, and I think I'm over a little bit, or quite a lot.
Thank you so much.
But I will, I will stop there, and I will ask Pramila to maybe moderate this session. And here we go, moderate the session to see if there are any questions in the chat, the Q&A.
Yeah, there's one question from Dr. Quadros. Is the gut distribution of bacterial effects dictated by regions such as ileum versus colon? And there's another question, does maternal gut microbiome affect the brain and subsequent behavior?
So the first question, we don't know about the regional effects in the small intestine. At least the transit time data suggested that the defect or the change is not in the small intestine, so we haven't looked carefully, and so we just don't have that data. And so we're focused a little bit more on the colon. And again, it's a new project, so there's still a lot of exploring we need to do, but the colitis data also suggests that there's a compromise in the colon. So I think that that's where we'll focus. The maternal microbiome of all animals, it is important, meaning that the microbiome of the mother is passed on to the offspring at birth, and that's the first exposure to live microbes and shapes the microbiome. There's a number of studies from many groups. I think Jun Hu, who spoke last year at this meeting at Harvard Medical School, has been doing some of the most pioneering work in the space in the past few years, I think, where he's shown that through various different inflammatory effects, the maternal microbiome affects behaviors and GI outcomes. He had a paper just last week on this topic, but of course, we and many others have worked on the effects of the maternal microbiome, and it appears again in animal models to be important in shaping both behaviors, GI function, as well as inflammation.
One more question, what is the age range of the mice in your study, and how does it correlate to human age, and do you look at changes longitudinally?
The age range of these animals, technically speaking, is we behavior test the animals at 7 or 8 weeks of age, which is adolescent, I think roughly correlated to adolescence in humans. And we are not trying to model the age component, though we can, and obviously it's an important variable in all of our experiments. We're using time points, that have been established for both the SHANK3 mouse as well as just various different behavior testing paradigms in all animals, and they show the effect there. But we haven't looked at earlier stages, which can be a challenge because animals have to develop to a certain point before they show these complex behaviors. So unless the animal is socializing, you don't know if there are changes in socialization until they're vocalizing. You don't know there are changes in vocalization. So there is a lower age range where we can't test the animals, but we haven't gone to the older animals. We haven't taken this particular paradigm to mice that were anything older than about 10 or 12 weeks of age. So we're still looking at very early stages of the animal's life.
Thank you very much. I think that's it for questions. So should we move on to the next talk.
We shall. Thank you very much, Pramila. And so I'll introduce the next speaker. The next speaker is Joseph Buxbaum. And the title of Joseph's talk